Hello everyone, welcome to my channel Anupma Biology Classes. In this video, our topic is respiration in plants. This video is taken from Plant Physiology series, CBSE class 11th, part 1st of chapter 15. Now, let's come on the lecture overview. So in this video, we will focus on some important topics like introduction of respiration, definition of respiration, types of respiration in which first is aerobic respiration, then anaerobic respiration, and then fermentation. After this, respiratory substrates, and the last one is adenosine triphosphate, that is ATP. So let's start the topic, in which first thing you have to know that what is respiration. So at first, introduction of respiration. Respiration and breathing are two separate processes. The gaseous exchange, that is intake of oxygen and release of carbon dioxide, is breathing. It should not be confused with respiration. The respiration occurs inside the cells. All animals, including human beings, breathe to live. Plants are different. They require oxygen for respiration to occur and they release carbon dioxide. However, they do not have a special organ such as lungs for the gaseous exchange. Even then, they have a kind of system which ensures the availability of oxygen to every living cell and consequent release of carbon dioxide into the atmosphere. For this purpose, they employ stomata and lenticels. In fact, these organs are not respiratory organs as they also perform gaseous exchange for photosynthesis and lose water vapors in transpiration. Plants do not have special respiratory organs because of the three possible reasons. So what are these possible reasons? First one is every plant parts take care of its own gaseous exchange needs so that there is very little transport of gases from one part to another. Unlike animals, the plants do not have a great demand for the gas exchange. It is because the rate of respiration in plant organs like roots, stems, leaves is far lower than that of animals. Moreover, the plants have an additional internal supply of oxygen and consumption of carbon dioxide by the process of photosynthesis that occur in all green cells. Plant organs are designed in such a way that their living cells are located close to the surface where atmospheric gases can diffuse easily. The leaves and other tender parts have stomata. Bark or woody parts of the thick stem and roots have lenticels. Moreover, the loose packing of parenchymatous cells in leaves, stems, and roots provides a system of intercellular spaces that facilitates free movement of gases inside the organs. Now, what is the main difference between the breathing and respiration? So, see here, breathing is a physical process involving exchange of oxygen and carbon dioxide, while the respiration is a biochemical process involving oxidation of glucose to carbon dioxide and water. Breathing here, energy is not released, while in case of the respiration, energy is released. In case of breathing, it takes place outside the cells, while the respiration takes place within the cells. In case of breathing, enzymes are not involved, while in case of respiration, respiratory enzymes are involved. So, respiration takes place all the time, while the breathing takes place also all the time. Respiration takes place in every cells, while the breathing takes place in the lungs. Respiration is the process that breaks down food to release energy, while the breathing is the process of gaseous exchange with the environment. Respiration uses oxygen obtained from breathing, while the breathing takes in oxygen from environment. Respiration produces carbon dioxide, while the breathing removes carbon dioxide produced during respiration. So this is all about the differences between the respiration and breathing. After this, see here the definition of respiration. So what is the definition of respiration? Respiration is in fact a complex process and no satisfactory definition for the respiration has yet been coined. Therefore, for working convenience, the respiration is defined as a process in which the respiratory substrates, So what are the respiratory substrates like sucrose, starch, glucose or other sugars, fats, organic acids, proteins, etc. are broken down through oxidation accompanying with the release of energy and its storage or conservation in the form of ATP with carbon dioxide release as a consequence. So here, the overall equation for the oxidation of glucose is as follow, like C6H12O6, that is the sugar, plus 6O2, that is oxygen. This forms 6CO2, that is carbon dioxide, plus 6H2, that is water, and ATP, that is 2870 kilojoule energy, which is usable. So, sugar and oxygen are the reactants, and the products are carbon dioxide, water, and energy. The above equation, that is 6H12O6 plus 6O2, which forms 6CO2 plus 6H2O plus energy, though misleading, summarizes the overall aerobic breakdown of glucose, releasing energy. The actual mechanism of respiratory breakdown is not a single reaction as shown in the above equation. It is rather the stepwise degradation of respiratory substrates consisting of many component reactions, each catalyzed by a specific enzyme. The released energy stored in the respiratory substrate is released slowly in the stepwise series of reaction. So, the energy released in the process is used up in various energy requiring process of the organism, and the carbon skeleton produced during respiratory breakdown of substrate is used as the precursors for biosynthesis of a large number of other essential plant products such as carbohydrates, proteins, fats, nucleic acids, pigments, cytochromes, etc. Now, do you know how many types of respiration? So let's see here. If we talk about the types of respiration, respiration is mainly of the two types, aerobic and anaerobic. In case of anaerobic, there are three types, that is alcoholic fermentation, lactic acid fermentation and the mixed acid fermentation. So this is the overview of the types of respiration. In case of all the respiration, types of respiration, at first glucose in the cytoplasm break down into the pyruvate. And here, in absence of oxygen, in case of yeast, it forms ethanol, carbon dioxide and energy. While in case of lack of oxygen in our muscle cells, it forms lactic acid and energy. While in case of presence of oxygen in mitochondria, it forms carbon dioxide, water and energy. So, cellular respiration in case of plants may be divided into two categories depending upon the availability of the atmospheric oxygen. And these two types are aerobic and anaerobic. So, in case of aerobic, we will see in the next videos glycolysis, tricarboxylic acid cycle, that is TCA, oxidative phosphorylation, and in case of anaerobic, we will see fermentation. So at first, see here the first type of respiration, which is aerobic respiration. So what is aerobic respiration? The oxidative breakdown of respiratory substrates with the help of atmospheric oxygen is known as aerobic respiration. It involves complete breakdown of substrates into carbon dioxide and water, releasing energy. Like for example, the simple chemical equation for the aerobic breakdown of glucose is glucose plus oxygen, which forms carbon dioxide, water and energy. So this energy is 686 kilocalorie or 2870 kilojoule. So this energy is released per molecule of glucose. Kilocalorie is equal to 1000 calories, and the kilocalorie is the amount of heat required to raise the temperature of 1 kilogram of water by 1 degree centigrade. Now calorie is intended to be replaced by joule. Now the second type of respiration is anaerobic respiration. So what is anaerobic respiration? Oxidation of respiratory substrates in absence of atmospheric oxygen is termed as anaerobic respiration. It involves incomplete breakdown of respiratory substrates in which the end products such as ethanol or lactic acid are produced and carbon dioxide is released. So you can see here, in case of anaerobic respiration, the energy released by anaerobic respiration is considerably less than the energy from aerobic respiration. Anaerobic respiration takes place at some stages in the cells of most living organisms. For example, our own muscle resort to anaerobic respiration when oxygen is not delivered to them fast enough, like when you do exercise. So certain tissues of the higher plant can carry on breakdown of respiratory substrates, particularly glucose, even in the absence of oxygen and conserve a small amount of energy. Such organisms which perform anaerobic respiration for some specified period are called facultative anaerobes. In this picture you can see the facultative anaerobes. They however switch over to aerobic respiration as soon as oxygen become available. Tissues of higher plants generally survive for few hours in absence of oxygen. This condition is known as anaerobiosis. In fact, some plants are subjected to anaerobiosis during certain stages in their life cycle. For example, certain seeds, when immersed in water during their germination, undergo anaerobiosis. So in case of anaerobiosis, lac cytochrome cannot use oxygen as hydrogen acceptor. Most lac catalases and peroxidases contain flavoproteins, so in the presence of oxygen produce H2O2, which is toxic. Some lac enzyme superoxide dismutase, so many killed peroxide and toxic radicals. Enzyme like the fumarate reductase must be reduced form to work. So you can see here the main difference between aerobiosis and anaerobiosis. In case of aerobics, require oxygen, more ATP of food, slower rate, lower food consumption and stamina. While in case of anaerobiosis, does not require oxygen, less ATP of food, faster rate, greater food consumption and burst speed. So these are the main differences between aerobiosis and anaerobiosis. After this, now the difference between the anaerobic and aerobic respiration. So what are the main differences? See here. Aerobic respiration takes place in the presence of oxygen, while the anaerobic respiration takes place in the absence of oxygen. Aerobic respiration, here the complete oxidation of glucose takes place, while in case of anaerobic respiration, the glucose molecule is incompletely oxidized. In case of aerobic respiration, end products are carbon dioxide and water, while in case of anaerobic respiration, the end products are either ethyl alcohol or lactic acid and carbon dioxide. In case of aerobic respiration, lot of energy is liberated, which is 38 ATP, while in case of anaerobic respiration, relatively small energy is liberated, which is 2 ATP. In case of aerobic respiration, it occurs in plants and animal cells, while in case of anaerobic respiration, it occurs in many anaerobic bacteria and human muscle cells. Now here you can see the formula, that means equations of the aerobic respiration and anaerobic respiration. So in case of anaerobic and aerobic, reactants are in case of anaerobic glucose, while in case of aerobic glucose and oxygen. If you talk about the combustion, in case of anaerobic it is incomplete, while in case of aerobic it is complete. In case of energy yield, in case of anaerobic, low energy yield, that is 2 ATP, while in case of aerobic, high, that is 36 to 38 ATP. Products are, in case of anaerobic, lactic acid in animals and ethanol and carbon dioxide in yeast, while the carbon dioxide and water is the end products of the aerobic. Location of anaerobic is cytoplasm, while the aerobic is cytoplasm and mitochondria. Stages in case of anaerobic are the glycolysis and fermentation, while in case of aerobic, glycolysis, link reaction, Krebs cycle and electron transport chain. So these are the main differences between the aerobic and anaerobic respiration. After this, now fermentation. So what is fermentation? Many microorganisms such as fungi and bacteria can grow and flourish in the complete absence of oxygen. Such microorganisms can utilize sugar to convert it into ethanol and evolve carbon dioxide. The overall equation for fermentation, as described by the French chemist Gay-Lussac in 1815, is, here you can see that, sugar plus yeast, which forms the alcohol and carbon dioxide. So, the process of fermentation is the form of anaerobic respiration carried by some microorganism. The fermentation can literally be distinguished from anaerobic respiration, that it may occur outside the living cells by the extracellular enzymes secreted by some microorganism. Previously, it was believed that the process of fermentation was strictly a chemical process, but Pasteur established that it is directly associated with the living organism. So, Pasteur in 1870 described that fermentation is directly associated with the growth, the metabolism of living matter. Pasteur further observed that the rate of fermentation was considerably higher under anaerobic condition as compared to aerobic condition. The inhibition of anaerobic breakdown of sugar into carbon dioxide and ethyl alcohol due to the presence of oxygen under aerobic condition was termed as Pasteur effect. This effect was further confirmed by Meyerhoff and Warburg. In the year 1897, the German chemist Buchner, while attempting to preserve yeast extract in the sugar media, demonstrated that a cell-free extract of yeast converted glucose to ethanol and carbon dioxide. The yeast extract was first analyzed by Harden and Young in 1905. The two scientists discovered that yeast juice loses its activity when it is dialyzed. It was later known that the yeast extract contained an enzyme that is known as gymase, which is non-dialyzable, and a coenzyme which is dialyzable. It is now well known that the gymase is a complex mixture of many enzymes and that several coenzymes are necessary for their function. Now, after this, if we talk about the types of fermentation, the first is alcoholic fermentation. In case of yeast, alcoholic fermentation occurs in some fungi like yeast and higher plants under anaerobic conditions. Pyruvic acid is broken down to ethyl alcohol and carbon dioxide in two steps. So here you can see the first step, that is the pyruvic acid is the first of all decarboxylated to acetaldehyde in presence of enzyme pyruvic acid decarboxylase or pyruvate decarboxylase. So in this fermentation, the first is conversion of pyruvate into the acetaldehyde, and after that the conversion of acetaldehyde, which is reduced to the ethyl alcohol by NADH and H plus, produced in glycolysis in presence of enzyme alcohol dehydrogenase. So, in case of alcoholic fermentation, first pyruvate is converted to acetaldehyde and then acetaldehyde is converted into ethanol. Now, the second type is here lactic acid fermentation, which is occur in bacteria. So, alcoholic fermentation occur in yeast and lactic acid fermentation occur in bacteria. Lactic acid fermentation is also known as homolactic fermentation. The breakdown of pyruvic acid yields lactic acid as an end product. The process occurs in many bacteria like lactobacilli, bacilli, streptococci, clostridia, etc. and in the muscle glycolysis. The pyruvic acid is reduced to lactic acid by NADH plus H plus in presence of enzyme lactic acid dehydrogenase. So in this case, conversion of pyruvic acid into lactic acid. So this is the process of lactic acid fermentation. Now, what are the differences between the alcoholic and the lactic acid fermentation? In case of alcoholic, the mechanism involves glycolysis, decarboxylation and reduction, while in case of lactic acid fermentation, the mechanism involves hydrolysis, glycolysis and reduction. Alcoholic occurs in bacteria and some fungi like yeast, while the lactic acid fermentation occurs in the lactic acid bacteria and muscle cells of the higher organism. Alcoholic fermentation, carbon dioxide and ethyl alcohol are the end products, while the lactic acid fermentation, lactic acid is the end product. In case of alcoholic fermentation, initial substrate is glucose, while in case of lactic acid fermentation, initial substrate is lactose. In case of alcoholic fermentation, the process is intracellular, while in case of lactic acid fermentation, the process is extracellular or intracellular. So these are the main differences between the alcoholic fermentation and lactic acid fermentation. So if we talk about the energy yield in fermentation, fermentation yields only about 5% of the energy obtained by aerobic respiration. This small amount of energy is sufficient to maintain the life of organisms such as yeast, many bacteria and other anaerobes. Vast majority of organisms are however aerobes, that is need oxygen for the respiration. Fermentation energy is too little to maintain the life of these organisms. They die within minutes in the total absence of oxygen. Fermentation can supplement the aerobic energy in them. Now, after this, see here the respiratory substrates. So, what are the respiratory substrates? Like animals, the plants also require energy for the growth, development, metabolism, and other life activities. This energy mainly comes from respiratory breakdown of various substrates. These substrates are called organic nutrients and synthesized inside the plants by the anabolic process. These organic substances, which are present in large amount in plant cells and respire completely to carbon dioxide and water, are called respiratory substrates. Under natural condition, only carbohydrates are oxidized. This respiration is called floating respiration. While if the carbohydrates are used up and its storage become acute, other protoplasmic substrates may also be catabolized, and this is termed protoplasmic respiration. Now, if we talk about the respiratory substrates, organic nutrients which are synthesized inside the plants by the anabolic process, respired completely to carbon dioxide and water, under natural conditions, only carbohydrates are oxidized in case of floating respiration. And if carbohydrates are used up and shortage becomes acute, then other protoplasmic structures may be catabolized. This is known as protoplasmic respiration. So here you can see the interrelationship among the metabolic pathways, showing the respiration mediated breakdown of different organic molecules to carbon dioxide water. So this is the oxidative degradation pathway of various respiratory substrates like the fats, carbohydrates and proteins. So as first, see here the respiratory substrate is usual respiratory substrate is glucose. Some cells can only respire glucose such as brain neurons. Other cells can oxidize lipids or amino acids. So glucose is a monosaccharide hexose molecule which acts as the chief respiratory substrate in plant catabolism. Second is starch in case of carbohydrates. Starch is the chief polysaccharide reserve food in plant cells. The starch is broken down to glucose 1-phosphate units by the action of enzyme starch phosphorylase. The glucose 1-phosphate is further converted to glucose 6-phosphate by enzyme phosphoglucomutase. The glucose 6-phosphate is an intermediate of glycolysis and oxidized completely to carbon dioxide and water. Now sucrose. It is the principal soluble disaccharide which moves from cell to cell and reaches wherever it is needed. The sucrose is converted to glucose and fructose molecules by the action of enzyme invertase. After this, see here the fructose. Fructose is also a hexose monosaccharide respiratory substrate. It is directly converted to fructose 6-phosphate by enzyme fructokinase and then it enters into the glycolysis. So in case of carbohydrate, starch, sucrose, glucose and fructose are the respiratory substrates, but the main respiratory substrate is glucose. Now, if we talk about the fat as a respiratory substrate, When fat acts as a respiratory substrate, they are first converted to fatty acid and glycerol. In presence of enzyme lipase, these fatty acids and glycerols may be converted to sucrose in germinating fatty acids, or they may be oxidized to carbon dioxide and water. Glycerol is phosphorylated to glycerol 3-phosphate in presence of enzyme glycerol kinase. So the fatty acids are converted to acetyl coenzyme A by the beta oxidation. The acetyl coenzyme A enters into the Krebs cycle and oxidizes to carbon dioxide and water. Now, in case of organic acids, under certain circumstances, the organic acids may be used up as a respiratory substrate in some tissues. For example, malic acid accumulates in dark in some succulent plants. This malic acid under a continuous dark period for a longer time is required to release carbon dioxide and water. Some plants such as oxalis, spinach, etc. accumulate oxalic acid probably for the oxidation of glycolic acid in presence of enzyme catalase. The oxalic is respired to carbon dioxide and water. Now, in case of proteins, the proteins are made up of varying number of different amino acids. The proteins are oxidized when shortage of carbohydrate become acute. Proteins are first broken down to amino acids by the action of proteolytic enzymes. These amino acids are converted to non-nitrogen containing organic acids by deamination reaction. So these organic acids enter into the Krebs cycle are further catabolized to yield energy. So protein is first converted into the amino acid, then amino acid deamination to form organic acids and ammonium salts, and these organic acid used in the Krebs cycle to form carbon dioxide and water. So here you can see the main respiratory substrates are the carbohydrates, in which the simple sugar, that is glucose, fats here, fatty acid and glycerol, and in case of protein, which is broken down to amino acid. So these are all about the respiratory substrates. After this, what is ATP? That is adenosine triphosphate. You know that adenosine is known as the energy currency of the cell. So what is this? The energy releasing process in a cell involves oxidation and reduction reactions. During oxidation of respiratory substrates within a cell, the energy contained in them is released in a series of slow stepwise reactions controlled by respiratory enzymes. All the energy contained in a respiratory substrate is not released free into the cell. It is trapped as a chemical energy in the chemical bonds of organic phosphates for use when needed. These energy-rich compounds are called energy carriers. The most important energy carrier is adenosine triphosphate or ATP. It carries energy in the two terminal phosphate bonds. The formation of these bonds require a large amount of energy used because they are formed against a force of repulsion between similarly charged radicals phosphate and phosphate. That means phosphorus and phosphorus. Hence, these bonds are known as the high energy or energy rich bonds. These bonds are shown in the chemical formula by the symbol, that is AMP, adenosine monophosphate, attached to the 2-phosphate molecules. So, high energy bonds are unstable and readily break down by the addition of water, releasing a correspondingly large amount of energy and freeing inorganic phosphates. The energy released by ATP is used by the cell in its chemical reaction and is called the biologically useful energy. Cellular respiration thus primarily serves to create high energy chemical bonds in energy carriers. ATP is mobile and transports and chemical energy to the energy consuming process anywhere within the cell. However, the ATP molecules do not pass from one cell to another. So, biological oxidation may release about 114.5 kilocalorie of energy per molecule of oxygen reduced, and this energy can produce 6 high energy bonds of ATP. So removal of terminal phosphate group of ATP by the hydrolysis yields ADP, a free phosphate group, and 7.3 kilocalorie of energy per molecule, and separation of the terminal phosphate group of ADP by another hydrolysis gives AMP and 7.3 kilocalorie of energy per molecule. So, equal amount of usable energy are released per molecule of ATP or ADP hydrolysis. Free energy of 7.3 kilocalorie per molecule is released under standard laboratory conditions in a test tube. Under typical conditions inside cells, hydrolysis of ATP to ADP release about 13 kilocalorie per molecule energy. ATP was discovered by Carl Lohmann in 1929. So, who discovered ATP? Carl Lohmann in 1929. Its formation and breakdown by union and separation of high energy phosphate was described in 1941 by Fritz Lippmann, a German biochemist in USA and Nobel laureate. Lippmann also proposed the sign for a high energy bond. So this is all about the adenosine triphosphate. So this is the end of video in which we knew about the introduction and definition of respiration, types of respiration, respiratory substrates, and adenosine triphosphate, that is ATP. So the next upcoming video will be on the glycolysis. So if you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.